All right, in this video, I'm going to go through the idea of something called codominance, which is another kind of complication of the dominant and recessive ideal that we have from Mendel. And we're going to end up by following a codominant trait through a pedigree. All right, so a good example for codominance is the ABO blood typing system in humans. All right, so you probably maybe you have been blood typed before. You know whether you're type O or A or AB. If you haven't been blood typed, one way you can find out is by donating blood. They'll type it for you and let you know. So there are many different ways to type blood. ABO is only one of them. And this is separate to the positive and negative typing that you get. So, sometimes you'll get, someone will tell you, I'm type O negative or I'm B positive. Now, whether you're A, B, or O has no relation to whether you're positive or negative. Those are two separate inherited traits. So we're not talking about positive and negative in here. We're talking about ABO. All right. So what these ABO blood types are looking at is what kind of decorations or glycolipids you have studding the surface or decorating the surface of your red blood cells. So if you think back to a couple of units ago, we talked about cell membranes and how cell membranes are coated with things like receptors and proteins and glycolipids and glycoproteins. One such glycolipid is shown here. So if we look at this, these molecules, there's 1, 2, 3 of them drawn here. If you think about the structure of a cell membrane, this squiggly bit here, this is a non-polar hydrocarbon tail. It's the lipid part of the molecule. And that's the part that can embed down in the phospholipid bilayer. And then this part here is the glyco part of the glycolipids. This is the glyco part, which means sugar, glyco. And this is the lipid part, the lipid tail, right? So the glyco part is made of different sugar rings. So if you think about, we learned about glucose, ribose, galactose, all these different monosaccharides, and you can chain them together to make disaccharides, polysaccharides. So this is a short chain of 1, 2, 3, 4, 5 with a little branch, 6 sugars kind of chained together to make a short chain and then stuck onto a lipid tail. And that's what makes this a glycolipid. In humans, we have the ability to make these 3 types of glycolipids that can be decorated or put on the surface of our red blood cells. And you can see they're slightly different to each other. So we'll start from the bottom and go up. This one here, you can see has the tail that goes in the membrane. And then this short chain here, and then a little L-shaped piece here, a little branch at the end, right? And this is the glycolipid that we're going to call O. It has a name O, right? The letter O. This is our O antigen or our O glycolipid. Here's the B antigen up here. It has the glycolipid tail. It has the same structure here as O, but it has this extra thing stuck on the end, this extra little sugar. All right, so the only difference between this one and this one is this end piece. And that's what makes this one B. It's different, so it has a different name. And then the top one here, the A antigen, again, it has the same basic pattern, just the same. You see that in that one and this one. But instead this has another sugar stuck on the end, and it's just a different sugar. Can you see how this one is pale blue and this one is dark blue, and this one there is nothing there? And that's what makes the difference between the O antigen, the B antigen, and the A antigen. They're all very similar. They're all just these decorations. And the only difference between O, A, and B is the very end of the chainy bit that sticks up. Does it have an extra bit sticking up? And if it does, what is it? Is it a dark blue sugar or a light blue sugar? They're not colored in real life, but you get the idea. Different molecules sticking out. Okay, so the ability to, whether or not you can make these three molecules is controlled by a gene, and we call that gene the I gene. All right, the I gene, we give it the letter I. And the ability to make the A glycolipid, we say is this allele, IA, this allows you to make this molecule. So here we say glycolipid A, which is right here, is made by this allele, capital IA. All right. Then there's another version of this gene, and it looks like this. We call it IB, capital IB, and this is another version of the same gene, but instead this gene makes the glycolipid look like this. So glycolipid B, which is this one, is made by this version of the I gene, capital IB. And then this version of the glycolipid at the bottom is made by a version of the gene that is little I. Now, you can see here I've used capital and lowercase letters very strategically, and that's because in this system, capital IA and capital IB, these are what we call codominant to each other. They're codominant. And this one here, this is recessive, and that's why I've used the lowercase. All right, so we're used to thinking of just two alleles. One is dominant, one is recessive, and they work together like the flower colors in Mendel's experiments. But this is slightly different. There's not just two alleles, there's three. And that's okay. They can be 5, 6, 7 alleles of a certain gene. I mean, it just depends on the gene you're looking at. It just happens that in this case, there are 3 versions of this gene that you find in humans: big IA, big IB, and little I. And each one can help you make a protein that can make one of these glycolipids. All right, so let's put this into context in terms of blood type. So this diagram here on the far right, the dark red circle, the big one, this is supposed to be a red blood cell, right? My fabulous artwork. So red blood cell, red blood cell, red blood cell. All right, and then you can see on the surface of the red blood cell what's decorating it. And I'm using this red circle to represent the A antigen, the A glycolipid, this purple square to represent the B glycolipid, and this black bar to represent the O glycolipid, right? So if we were to put that in, look over here, this red blood cell is only decorated with A glycolipids. This blood cell is only decorated with B glycolipids. And this red blood cell is only decorated with O glycolipids. All right, so this person, if they went to take a blood test, it would come back and say they are type A blood. This person would come back as type B blood, and this person would come back as type O blood. That's what the blood typing is looking for. It's trying to detect which of these decorations do you have on the surface of your red blood cells. All right, so that's kind of the background of the scenario. Let's go on and look at the next slide. So same pictures again. Here's my red blood cells from different people. So as we just said, this person would be told they have type A blood. This person would be told they have type B blood, and this person would be told they have type O blood. That's the phenotype. Remember, the phenotype is how you would physically describe what's going on. It's kind of telling you what decorations do you have. Now, in real life, you can't look at the red blood cells and see what decorations are there. But in this diagram, I'm kind of faking it and showing that you can. And so if we look at this red blood cell right here, the only kind of decorations you can see on its surface are the A glycolipids, the red circles. And the genotype here, this person must have a big IA allele and a big IA allele, because all they're making is the A decoration, the A glycolipid. This person here, number 2, all they're making is B decorations. Their red blood cells have nothing but purple squares on them. So this person is big IB, big IB. And this person here is just making O decorations. And so the O decorations are made by this little I allele. So this person is And the reason I gave everybody 2 alleles, like two here, 2 here, 2 here, is because humans are diploid, right? We have 2 of every gene, 2 of every chromosome, so therefore 2 of every gene. And so those would be the genotypes of these people. All right, well, that seems pretty straightforward, but we have some other scenarios we haven't thought about yet. So let's move to the next slide. So here's someone who, if we were to look at their red blood cells with this magical microscope that can actually see the decorations, which doesn't exist. There's no such microscope that can do that. But if it could, we would see on this person's red blood cells, there's both A decorations and O decorations, right? So this person is making A decorations and they're making O decorations. And so that would be their genotype. However, we've said already, and we can show by the letters we're using, that IA and IB are codominant, but codominant with each other. And they're both dominant compared to little I, which is recessive. And if you remember, when you have a heterozygote here, which has a dominant and a recessive, like this is the dominant one and this is the recessive one, you only see in the phenotype the dominant allele. So this person would have type A blood. Even though they've got this allele, and even though it's showing up on their red blood cells, we wouldn't be able to detect it or see it with the way we look at blood. And so this person would come back with type A blood, just the same as this person. So someone who is type A blood could be like this, or they could be like this. We have no way of knowing. All right. It's like those purple flowered plants that Mendel had. They could be big P, or they could be big P, little p in terms of purple flower gene, but we had no way to tell them apart. So let's do the same idea here on the second one. This cell is coated with B antiglycolipids and O. So this is their genotype. But if they had got a blood typing done, it would come back and say that they were type B blood, because you can't see, the little I is not appearing in the phenotype. And then the last one here, we have this cell is decorated with both type A and type B. So we would write their genotype like this. And we would say that their type, this would be type AB. It's quite rare actually to have type AB blood, but there are some people that are type AB. All right. So if someone is type AB, they've got to be this. All right. If someone is type O, they've got to be this. But if someone is type A or type B, we can't tell between this one, this one, this one, or this one. It could be any of those because the phenotype doesn't reveal the exact genotype. All right, and so this is what we call codominance, where in this example right here, where both of the alleles are together, the dominant alleles are together, A and B, you're seeing both A and both B at the same time. One doesn't appear instead of the other in the heterozygote. You see A and B at the same time. This is different to incomplete dominance. In incomplete dominance, which was the example with the flowers that turned pink when you had red and white parents, there the red and white appear to us to have blended together to make a new color, pink. Just really to do with how our eyeballs detect it. Really, red and white are both there. So it's really kind of the same as codominance, but our eyeballs make it look like it's blended, and pink is halfway between red and white. But in codominance, there is no halfway between A and B. It's either A or B or A and B together. There's nothing blended here. There's both A totally present and B totally present. And that's why we call it codominance, and that's why it's slightly different to incomplete dominance. It's not blending. It's both at once, and you see them both. All right, so that is how the human blood typing works. Let me show you an example in a pedigree so we can see how you might figure this pedigree out and try and determine people's blood types. And you can do this for yourself at home. I'm going to once again use my family tree. So I'm going to have, here's my mom and here's my dad. If you don't know what I'm drawing here, this is a pedigree, and there's another video in the genetics playlist that explains to you how this works. All right, oops, sorry, lost my pen for a second. All right, here's my mom, and then here's me, and then here's my brother. Okay, so I am blood type B. All right, that's my blood type. So, you know what? I'm going to start this over higher up. So here's my mom, here's my dad, here's me, here's my brother. I am type B. All right, so if I'm type B, what are my possible genotypes? My possible genotypes would be I big B, I big B, or I big B, little I. I can't tell right now. All right, here's my brother. He is type O. All right, so if he's type O, we know what he must be, right? He's got to be 2 little i's. That's the only way you can be type O. My father was also type O. All right, so we also know his genotype. It has to be type little I, little I. O's are really easy because there's only one way you can be type O. Now, because of that, we can tell something about me, right? Think about where I got my alleles from. I got one from my mom and one from my dad. So what did my dad have to give me? A little I or a little I. That's all he had. Doesn't matter which one he gave me, it would have been the same. So I cannot be this because this requires that one of my parents gave me a big IB and the other gave me a big IB. And my dad hasn't got a big IB to give. So I can't be that. So we can now say that I've got to be big IB, little I. All right, that's definitely me. My mom is also type B. All right, so she's type B. What are her options? She could be big IB, or she could be big IB, little I. That would give her type B, either of those. But we can determine her too, because look at my brother. For my brother to have got 2 little i's, who did he get them from? He got one from one parent and one from the other parent. Well, we know he must have got a little I from my dad because my dad only has little i's, so that makes sense. But he must have got his other little I from his mom. So if we go up here, that means mom must have had a little I to give him, so she can't be this. So we can get rid of this, and we know for sure she's that. So my family tree actually works out pretty nicely here for figuring out blood type, genetics. Sometimes it works out easy. Sometimes you can't figure it out. Sometimes there's not enough information. Now here's another one for you. My husband is type A. All right. All right. So he is either big IA, or he could be big IA, little I, right? Those are 2 options for him. I do know that his mother was type O. So knowing that my husband's mother was type O, which means his mom was 2 little i's, that means she must have given him a little I. So we can cross this one out, right? We know he can't be this. So we know he's got to be big IA, little I, all right? And we have a daughter, and she's not been blood typed yet, so we don't know what she is, but what could she be? Let's see. So I am big IB, little I, that's the mom. He is big IA, little I, that's the dad. So all the possible options that we could have a big IA, big IB, child. We could have a big IA, little I. We could have a big IB, little I, or we could have a little I, little I. So my daughter actually has a 25% chance of any of the blood types. She could be AB, she could be A, she could be B, or she could be O. I don't know. I saw some point I'll find out, but I'm quite intrigued. But that's kind of how you work these blood type genetic problems through. Hope that's helpful.